Welcome, everyone. As your lead medicinal researcher, I am absolutely thrilled to welcome you, our incoming college students, to your orientation for the computational lab. Today, we're diving into an explainer that is frankly going to completely change how you view chemistry and biology. We're exploring the dynamics of discovery, specifically molecular simulation in modern medicine. Look, if you're used to staring at those standard static plastic models of molecules from your textbooks, get ready, because we're about to blow your minds. Okay, let's jump right in. So here's our quick syllabus for today's orientation. We'll start with number 1, beyond static models. Then 2, the physics engine. 3, molecular vital signs. 4, modern drug discovery. And finally, 5, the AI future. All right, section 1, beyond static models. For decades, scientists really had to rely on static molecular structures. It's kind of like looking at a still photograph. It captures just a single, frozen moment in time. But here's the thing, biological molecules aren't frozen plastic toys. They are incredibly dynamic. A static image just doesn't tell us how a protein wriggles around, how it breathes, or how it actually reacts in the real world. So on the right here, we have molecular dynamics, or MD simulation. If that static model is a photograph, an MD simulation is basically a high-definition video. It shows us exactly how these proteins, ligands, and biomolecules dynamically behave under actual, real-life physiological conditions. To give it a formal definition, molecular dynamics simulation is a computer-based technique that predicts dynamic molecular movement and interactions at the atomic level over time. Really, this is our ultimate tool for observing how biomolecules actually behave out in the real world. We use this to see exactly how proteins fold, or how drugs physically dock into their target sites. We aren't just guessing anymore, guys. We are predicting actual atomic movement using massive computational power. Moving on to section 2, the physics engine. Force equals mass times acceleration. F equals ma, Newton's second law of motion. I know you all remember this one from high school physics, right? Well, it might seem totally wild that the primary engine driving modern drug discovery and structural biology is the exact same equation we use to calculate the trajectory of a falling apple. But it's absolutely true. In our simulations, force literally determines the motion of every single atom in the system. And this gives us what we call a cyclical physics engine. Step 1, we just apply Newton's laws of motion to our molecular system. Step 2, we calculate the forces between the atoms. But the real magic happens in step 3. We do this repeatedly over extremely tiny time intervals. We're talking fractions of a billionth of a second. By calculating how atoms push and pull on each other over and over and over again, we can predict continuous molecular movement. We are literally watching the atoms dance by solving F equals ma millions of times a second. Okay, section 3, molecular vital signs. Let's break down the diagnostic metrics you're going to be using pretty much every single day in this lab. These are the parameters we analyze in MD simulation to validate our molecular docking and study conformational changes. First up is RMSD, or root mean square deviation. This evaluates structural stability. Think of it like a patient's core vital signs. If the RMSD is wildly fluctuating, well, our protein is unstable. Next is RMSF, root mean square fluctuation. This one measures residue flexibility. Imagine leaves blowing in the wind, right? The trunk is stable, but the individual leaves flutter. RMSF tells us exactly which specific parts of the protein are fluttering the most. Then we have the radius of gyration, or RG. This determines the compactness of proteins. Imagine an ice dancer pulling their arms in to spin faster. That creates a tighter radius of gyration. It basically tells us if a protein is folding up tightly or unraveling. We also analyze hydrogen bonds to study interaction stability. Think of these as hands holding tightly between different parts of a molecule, keeping it locked into a functional shape. And finally, we step-by-step analyze SASA, which is the solvent accessible surface area. This is basically like measuring exposed skin. We calculate exactly how much of the protein's surface area can actually be touched by surrounding water molecules. Why does that matter? Well, if a drug target site is hidden deep inside and has a really low SASA, our drug just won't be able to reach it. Now, to calculate all those complex vital signs, we rely on some serious computational muscle. Here are the heavy-lifting, industry-standard software tools we use: GROMACS, AMBER, NAMD, CHARM, and Desmond. You are going to become very, very familiar with these programs. They are the software environments that actually execute those millions of Newton's law calculations. Which brings us to section 4, modern drug discovery. Now, you might be wondering, why spend massive computational resources on these molecular simulations? And it's a completely fair question. Running GROMACS or AMBER for weeks on supercomputers requires immense computational resources. It is definitely not cheap to run these servers. So, why do we do it? Why spend so much time in computing power just simulating atomic wiggles? Simply put, because it replaces slow physical experiments, significantly reducing both laboratory costs and time. Think about traditional wet labs. They require synthesizing thousands of chemicals, testing them in test tubes, failing, and trying all over again. That takes years and billions of dollars. MD simulation gives us atomic-level insights entirely digitally. We can identify stable drug-target interactions and optimize lead compounds way before we ever pick up a real-life test tube. That saves massive amounts of research time and cost. It's a real game changer.
And the real-world applications here are just staggering. We use MD simulation across the board in drug discovery and development to design effective drug candidates. In cancer research, we use it to investigate inhibitors targeting cancer-related proteins, trying to lock them down and actually improve patient outcomes. It's heavily utilized in antiviral research, too. In fact, these exact simulations were widely applied in studying viral proteins and antiviral drug candidates during the SARS-CoV-2 pandemic. It fundamentally aids in protein folding studies to help us understand how proteins form functional 3D structures. And it's absolutely vital for vaccine and biotechnology research, supporting studies of biomolecular stability and antigen-antibody interactions. By simulating how an antibody physically binds to an antigen, we can develop far more effective vaccines.
All right, last section, Section 5, the AI future. The field of pharmaceutical sciences just continues to grow at a breakneck pace. And this equation right here, this is the future. AI-driven drug discovery plus molecular dynamics equals unprecedented therapeutic innovation. Artificial intelligence and bioinformatics are advancing so incredibly fast. When we merge AI's ability to spot massive data patterns with MD simulation's ability to validate precise atomic-level biomolecular mechanisms, well, we accelerate rational drug design and precision medicine like never before. So, as we wrap up today's lab orientation, I want to leave you with a question to chew on. As bioinformatics and these simulation tools remain absolutely critical for understanding biological systems, what complex biological system will you simulate to discover the next life-saving breakthrough? The tools are here, the physics is ready. Now it's time for you to start simulating.